And there it started. So this is what I sent you today, and I probably will have some more materials to add to this. And oh yeah, and so and I said for the test that we're going to have on the Wednesday, 2 weeks from today, I'll make sure I get that review report. I got to try to get that tomorrow or the next day. Let's see. She has. I have assignments I have to carry out. Leaves. Do you know how much it takes to remove a giant oak tree? We have. I don't want to think about it. I do like them, but they're just huge. Okay, here we go. So basically, the muscular system is a fascinating, and the only good part, at least of having these close together, is they reinforce one another. Because what will happen is we're going to basically, when we talk more about nerves, we're talking about the neuromuscular junction. Again, except rather than being a neuromuscular junction, we're talking about another synapse. We're talking about nerve-to-nerve junctions. They work very, very similarly. So understanding the way the nerve works, whether it's talking to another nerve or it's talking to a muscle, really is there's so many similarities. I think it helps to reinforce each other. And that's why, and not just me, we all do them basically in this order. This is a pretty consistent pattern that you begin to see. And because what all this is, there's two major control systems. There's the nervous system and there's the endocrine system. The nervous system is right now. We're talking microseconds, tiny fractions of a second, much faster than a muscular response. The fact that your mind is starting to put these parts in actions and generate a particular response. So it's very, very fast. The other control system is called the endocrine system. So if that's fractions of a second in the nervous system, then we're talking an endocrine is over time. Seconds, minutes, hours, days, whatever. Things that happen rapidly, that start rapidly, end rapidly. Things that start slowly, end slowly. Very consistent. You'll see that all the time. It's very rare that something starts rapidly, ends slowly. It just doesn't work that way. And so effectively, it's the idea of distinction. So you're on your way to home, on your way to work, you're on your way to school, you're in the car, you're going someplace, and you're driving, and all of a sudden some idiot, like this morning, a couple of cars in front of me trying to squeeze into a spot in the fast lane on I-79. Never a good, I figured it's over. And we've all done that. You make that super fast response, and thankfully nothing bad happened, and you're like, oh my, and you can feel it even as you're resuming driving. And get control and everything's moving around. You can feel your heart moving, you can feel pumping in your chest, you're increasing your respiration. Your ability to make that immediate maneuver, maybe hitting the brake, hitting this, pulling into a different lane, all of that nervous system. The prolonged effect that it has on you systemically is endocrine system. We'll explore that, the linkage between them, and one of the systems I'm going to talk about very shortly, that is the very last part that we do, called the autonomic nervous system. I touched on that a little because that's really what controls smooth muscles that are there. So the nervous system is the master controller, the master communicator, no question about it. And it's just like, very similar to how muscles work, nerves are electrical, and they communicate with whatever their target is by chemical, even if it's nerve to nerve. It's very, very few of them are not. There's very few that are electrical to electrical when influencing the nerve. Almost all the time it's release of chemicals. Those chemicals are what we're going to address in the third of these three power points. They're the neurotransmitters. We already have a leg up on the one that we have to know the most about. That's acetylcholine. The second of these two power points really delves into the physiology of how this all works. This is more maybe the first 3/4 of the anatomy lesson. Is the way I would describe it here. So this is the master controlling system. Very rapid, very specific, very immediate. Effectively. And this is intuitive. And the misconception is people think we have so many things that we're sensing our environment, and then so many things we do about it. That's really not true. We have basically we hear something, we see something, We decide what to do about it, like that car, and we take an action. So the sensory input, whatever the receptors are, maybe in the car it was visual. Maybe you're at the same time you're aware of what yours is doing. You're keeping an eye on the mirrors. You already checked this. You know how fast everybody's going. That's the sensory input. And then when you see that subtle change, now you're going to basically create you're going to effect an action, hence the name of an effector. The names that you will see are afferent, notice the way I pronounce it, and efferent. Efferent is really where it started. It comes from the word effector. And we use it because if you say afferent and efferent, they sound too much the same. When you're in a scientific field or a medical field, you gotta be specific about what you're saying. Okay, so that's why we'll accent a letter like abduction or adduction. You don't want to make mistakes that are easily avoidable. That's an example. So we have afferent sensory fibers. We have efferent effector fibers, activator fibers, and they basically target muscles and glands. Everything else we call interneurons, in between neurons. It might surprise you that maybe 98 to 99% of all of our nerves are the integration, the in-between nerves. I know we sense a ton of stuff, and I know we can act on it in a ton of different ways, but the big deal is processing. That's what it's all about, these interneurons. We process that information. If you're listening to me now, if you're doing, it doesn't matter. Students do something else or studying for another test, but it doesn't matter. But you're able to process a certain amount of information when you go back and listen to it. That's kind of what's going on. You're hearing it. How you decide to react to it is how things. There's so many ways to analogize this. Taking a test, you're looking at the questions, and basically, once you read the question, okay, and hopefully understand it, then how do I recall the information? What about this? What about this? And finally, you exercise a decision. Is it A, B, C, D, or E? After I've read and tried to understand what those are. So it works for everything that we do. It just works relatively rapidly in certain circumstances, and obviously in an examination environment, then you typically have more time to respond to that. Typically, not always. So you see something, you make a decision, I'm thirsty, you grab it. Easy. But the key, the take-home point is these in-between nerves are the ones that really are the elite aspects. We very rarely see this sensory one in between and motor circuit. Very, very rare. That's a simple reflex. If you think about the knee jerk, we call it a simple reflex. Taps the knee, stretches something, the knee kicks out, you know somebody tapped your knee. Basically that. There's not a whole lot of in-between steps. That's about as simple as it gets when you talk about a nerve circuit. And so definitionally, we have these things. We have what's called the central nervous system. Central nervous system, or CNS, in the very first lab, I am sure that you were in an A and B. They talked about the dorsal body cavity. You had the ventral body cavity. It had the diaphragm separated into the thoracic and abdominal peritoneal. I've taught that lab many more times than Dr. Robbins had, for one very good reason. I'm considerably older than Dr. Robbins. Probably about 20 years older than he is. And so understand, we've all done that lab. It all starts the same way. In the dorsal body cavity, you have exactly one thing, your central nervous system. And what's that? By definition, brain and spinal. So that's an important thing. Whenever I say it's an important thing to remember, it's likely going to be a test question. See, there. I like that. You get a mini gold star. It's like my grandchildren get, except that translates to cash, which I can't do for my grandkids. We have a word for that. Bribery. It's okay. Nobody did that for me. My dad said, do that. I know how to answer right away, Dad. It's a very scary guy. You were such a good guy. Brain and spinal cord, dorsal body cavity. And that's really where the interpretation is. Anything that sticks out of it, we call, is different. That's the peripheral nervous system. So brain and spinal cord, central nervous system. And I use the term CNS, it's a universal acronym, as is PNS, peripheral nervous system. Anything that protrudes, sticks out. It starts with cranial nerves and spinal nerves. Okay. And then they do certain things and end in certain places and integrate certain things. I know you had a certain amount of background with cranial nerves. I don't believe you had spinal nerves. I mean, I don't get anything out of this, nor should you. Again, it's divided in divisions. Sensory, notice they use the term afferent nerves. Somatic means it comes from the body. The best way to interpret that is it's not visceral. Visceral means internal organs. You don't get a lot of sensors from that. From your body, you get a tremendous amount of information. So anything but, I mean, if it's coming from your liver, your stomach, your small, large intestine, your lungs, it's very generic information. Something's going on there. Versus it's very specific a lot when it's your skin, your muscles, your joints. You immediately know exactly wherever your body is positioned. I mean, close your eyes. Everybody in the room knows whether their elbow is flexed on the right or the left, whether your leg is. We get that kind of information. It's just filed away in case we ever have to use it. That's somatic sensory. Visceral sensory, something that's very nonspecific. The motor, and as you can see, efferent is a bigger part division. It goes to the effector organs, which by definition are muscles and glands. Somatic means, again, anything but the internal viscera, the internal organs. By definition, the autonomic nervous system, our last chapter that we're going to look at, again, at the very last week of the class, that is the one that basically the motor division going to all of those organs. And you'll see it here. Somatic, as you can see, with voluntary conscious control of skeletal muscles, effectively what it is. And the autonomic, and you could substitute the word automatic for that if you wished. Okay, smooth muscle, heart muscle, glands. We don't have any control over it. It's involuntary. And you're going to see this is really where homeostasis occurs. We have the combination of two branches, parasympathetic and sympathetic. What you're all using now is parasympathetic. Basically, it's your body in more or less a rest position. Whereas when you're active, when you leave here, start to walk, go over wherever you're heading. If you're heading to a car, you're heading back to a dorm, you're heading to grab something to eat. And all of a sudden, the action or the sympathetic division kicks in. And together they work, they play a pivotal role in homeostasis. We'll save that for after Thanksgiving. Even tell you why having all that turkey makes you sleepy. Okay? People don't eat turkey at Thanksgiving. What are those classes? Oh, come on. In my house, we had tofurkey, too. Tofu turkeys. Vegans, you know. I love cooking that way. Moving on. And you'll see this chart a lot. We're not going to go over it. The big deal here are these guys we start with. The nervous system is not just nerves. It's a unique system. Every other thing that you're going to study, you talk about different kinds of connective tissue. Dense regular, dense irregular, loose irregular, lining tissue, simple squamous, simple clump, all that stuff, right? That applies everywhere in the body but your brain. Your brain, the central nervous system, moreover than just your brain, has its own connective tissue. They're called glial cells. Collectively, we call them neuroglia. That's why we call it histology when we study the tissues. Two principal cells. The nerve cell, we're really going to just briefly look at that. And the neuroglia cell, or glial cells. And glio means glue. So they're the glue that holds it together. Nerve cells are amitotic. Just like muscle, skeletal muscle, when you're born, that's all you got. They're damaged. You're not going to, you can't make more. Maybe you can improve what you have, but you can't make more. Nerve cells are the same thing. You're born with them. If they're damaged, depending on where they're damaged, they may or may not be able to be repaired. If they're damaged at the important part of the cell, and again, I'm going to use the word nucleus advisedly, where what you and I would call the nucleus of that cell, but that's the long term, we will get to that, then that cell is going to die, and you can't get another one. No more, thank you. You can't make it. Interestingly, that's the bad news. The good news about that is they don't become cancerous because they no longer undergo mitosis. So just like skeletal muscle cells, they're amitotic. When you hear about people with brain cancer, it's the glial cells. Oftentimes they'll say, oh, they have a optic nerve glioma. It's a non-specific term for where the optic nerve is. And you looked at that when you have an idea about that from cranial nerves, right sort of underneath, sort of back and underneath here. Very, very difficult. So, and most tumors are benign, thankfully, 90 to 95%, but they occupy space. That's the problem with the tumor when you're in a closed space. I mean, I have like a little lipoma there. I had it as long as I can remember. As an adult, 50 years. Doesn't matter. Plenty of space. You can get bigger. Who cares? But in your brain, limited space. And so what happens when tumors grow in your brain, because you don't have a hard boundary, as it grows, it pushes and squeezes the stuff and damages. So the fact that it's benign doesn't mean it's not deadly. Ones that are cancerous are even more deadly. So that's sort of what that is all about. So we're looking at the glial cells, and I'll write them out to show you the relationship. There's 4 that are in the central nervous system, and 2 only. So you only got 6. And of the 2 in the peripheral nervous system, they have a certain amount of similarities. We have what's called astrocytes. We have oligodendrocytes. You're going to see these names anyway. You have microglia, and you have ependymol. So, they're the ones 4 in the central nervous system. The astrocytes are similar to, not identical to, in function, to what are called satellite cells in the peripheral nervous systems. And the oligodendrocytes have a very, very similar relationship. They're basically identical other than how they're designed, with a very famous cell in the peripheral nervous system called the Schwann cell. Those are the 6 cells you need to know. I'm going to endeavor to explain them from here. Voila. Told you didn't have to write them down. Astro means star shape. Astrocytes. We use a lot of those names. It's like coronaviruses. Corona means a crown. So we use a lot of terms that appear in a lot of different places. Astrocyte, microglial cells, teeny tiny. I have no idea the definition of how they came in the world. We'll start with the astrocytes. They are by far and away the ones there are the most. They are really the linchpin of all the glial cells. So we'll hear terms here. And again, in a tumor, one of the ones you hear a lot about is called an astrocytoma. There's so many. It does a lot of things. Perhaps the most significant thing that it does involves something that does with the capillaries. Capillaries, when you get to an AP2, are the vessels where, when they go into tissue, is where the exchange takes place. Oxygen leaves, carbon dioxide comes back in. Nutrients leave, waste products come back in. It's where the bloodstream comes in intimate contact. And the capillaries are blood vessels that are tiny, and they're really just made out of lining cells. And the lining cells are held together, and they have little loose openings in them, so this stuff can get through. In the brain, those capillaries are sealed tight. You might remember the name tight junctions. Tight junctions mean watertight. So nothing with water or or products dissolved in water in solution can get through. They can't. In order to get through, they have to be materials that are compatible with fat. Call them nonpolar covalent, oxygen, carbon dioxide. If you know somebody who takes a medication for depression, if they take for eating disorders, if they take a medication for schizophrenia or bipolar, whatever they do, if they take a pain pill, if they take something to help them sleep, those medicines work because they can cross what's called the blood-brain barrier. The basis of the blood-brain barrier are these unique capillaries that don't allow water through them, or particles dissolved in water. So they have to be able to move through the membrane. What reinforces that boundary and is really the roadway, the passageway between the blood vessel to the nerve are the astrocytes. You'll see in a second. The astrocytes do a lot. They're part of development. They're supportive in nature. Okay? This is the exchange role, is what I was just talking about. They're part of development. When they talk about guiding migration of young nerves, you're born with those nerves. It's they're coming together and forming interconnections with one another. That's really what development is about. As you grow, you're learning as you're beginning to use more and more of your brain, and you create more and more passageways, and where nerve cells intersect. It's unlimited. We use a relatively small amount of our brain, comparatively. And as you use more, you have plenty. It's like you got a 10 terabyte hard drive, and you're using a couple of gigs if you want to use the computer analogy. We all do. It's not anybody in particular. We all don't use a lot. And what they play a role in, like landmarks. When a baby's born, they can't do anything. They're total care. Without it, they're not going to survive. And everybody gets so excited when certain landmarks. The baby can hold its head up. The baby can start to crawl. The baby can start to stand. The baby can start to walk. The baby can start to talk. All of those things. Those are all development of the nervous system. Those wonderful landmarks. You got your camera out. You're sending pictures to grandmas and grandpas like us. I'm inundated with them. Everybody is. Probably you run out of stuff to say. My granddaughter keeps sending us this cute as a button grandson. And it's like, how many heart symbols can you put on? Do you know what I mean? Oh God, he's super cute. How many times have I said that? Didn't mean that, Vanessa. He's a beautiful kid. So it responds. Also, it adapts to signals like nerve signals and neurotransmitters. It influences how nerves work, and it influences information. They're a busy ass cell. Only way to say it. So what happens? What I'm trying to describe, here's the blood vessel, here's the nerve. This is the go-between. See, it goes through the membrane of the capillary because there's no openings. It goes through the membrane and along the membrane of the astrocyte, and it goes into the nerve. Oxygen, nutrients, waste products, we'll just use this for example, come out. Carbon dioxide and waste products, again, go directly in because they can get through the membrane of the nerve, and they get through the membrane here, and they use the passageway here as a conduit, and they work their way back to the blood vessel. That's what's meant by the barrier. It's not as easy as just coming out here and magically, they can't go from point A to point B just like that. Can't do it. The microglial cells are little scavenger cells. They're really analogous to tiny, sort of non-specific white blood cells, but they're very tiny. They have a lot of different processes, and they're constantly, most of the time, if they're dealing with microorganisms, that person is in big trouble. The brain is not supposed to have viruses and bacteria running around. They're not. However, there's a lot of debris. Nerves are busy, okay? They're making waste products a lot. There's subtle amounts of damage, and it's picking up that sort of those waste products and debris. They look a little like this, not a whole lot. Just to know that they're microglia, their name tells you all, they're super tiny. The ependymal cells, this is something, and they didn't point this out when you guys did the sheep brain dissection. If you'd had the chance and you looked inside those ventricles, like the lateral third and fourth ventricle in the sheep brain, sometimes you'd feel like this little bits of like funny looking brown stuff. Those were part of where cerebrospinal fluid is produced. We'll get into cerebrospinal fluid. What happens is it comes from the plasma. It's not plasma itself. It's sort of, we call it a filtrate. So you take water and some of the elements of plasma, they're modified to become cerebrospinal fluid. And part of that modification occurs in a little nest of blood vessel that kind of sticks out into those ventricles. It's covered in ependymal cells. The ependymal cells contribute to it, and because they're ciliated, they move it around. Because it's like any circulation, it has to be moving. We make about a pint of cerebrospinal fluid a day, and we absorb a pint. We reclaim a pint, basically send the fluid back to the heart. It gets redistributed, and we start all over again. That's just the way it's sort of an efficiency matter of recycling everything that occurs. And so those central cavities, those ventricles, are where we see this. And unlike the blood-brain barrier, it's permeable. Stuff can get in and out. So they kind of give you the schematic like this. They're covering tissue of the brain, and they're covering those ventricles, and they're also covering these little tufts of capillaries that you're going to see. When we get into the brain itself, you'll even see little areas like little siphons that will basically, like overflow valves, that will take when the fluid's produced, bring it back, and it collects and goes back in veins and goes back to your heart. Again, everything is recirculated. And now we get the oligodendrocytes. The oligodendrocytes, they are all about this. Both the oligodendrocytes and the Schwann cells, they make myelin. Myelin is, you looked at that brain, you saw two types of structures, a lot of gray matter and white matter. White matter are all the long axons, the transmitting and receiving structures that basically send signals all over the place. They're white matter because they're encased in a material called myelin. Myelin is basically a cell membrane. It's fat and protein. It's unique because we only see it associated with the nervous system, and it is in multiple layers. And moreover, it's very, very protective. The more myelin you have, the faster the signal. The thicker the nerve and the more myelin, the faster it conducts the signal. It's a very unique material. One of the real tragedies is a disease that causes areas of this myelin to harden. A word for hardening is called sclerosis. This disease is called multiple sclerosis. It's an autoimmune disease. The acronym we use to describe it is MS. Unfortunately, it is more common than you think. Not everybody requires aggressive treatment, but the ones who are really bad with it, it's not a good thing. So that it relates to this myelin. It happens both in the central nervous system, principally these oligodendrocytes, but it also can affect the Schwann cells. This is what they look like. Again, very limited space up in your brain. You have millions of these nerves, these tiny, fine axons, these fine threads. They're bundled together. What holds them together, so you can see this is the oligodendrocyte, it's a cell, and this is all the myelin. And it isn't just 3, it could encase up to 60 individual axons. And it helps to organize them and sort of pack them in tight, yet still keep them protected. And then you have space. The spaces are called myelin sheath gaps. Now, they used to have a different name. So the gaps are how the signal is conducted rapidly. White matter and myelinated nerves, the signal literally seems to jump from gap to gap. In the old days, and depending on where you worked, or if you're going to be using this material or studying it more, whoever is instructing or you're working with will use the term that I learned. It wasn't that many years ago. They're called nodes of Ranvier. For a French scientist or doctor, nodes being those areas in between where it doesn't have the myelin, chief gaps, and the name of the person, Ranvier. I love saying it. Very French. It's fun. Do you know what vichyssoise is? Oh, we're having vichyssoise for dinner. Cold potato soup. Doesn't sound as good as vichyssoise. Oh, really? That sounds elegant. Cold potato soup with leeks and a little bit of parsley. You have to take a leek in the soup. Leek. Oh, God. Heathens. Soups are good cold. Gazpacho? Oh. I'm going to work on your palate, Chris. Kristen. Got to work on it. It's like, it's made with cucumber and tomato and sometimes sweetened with watermelon and spicy, if you like it that way. Oh, it's delicious. It's Mexican. It's like it's a soup to use for leftovers. Okay. So the satellite cells, we think, that's why I put the question marks there, act like astrocytes. These are the peripheral nervous system, the nerves going out of your spine or your brain. So they surround the bodies, and you're going to see what we mean when we see cell body in a second. And they're similar to astrocytes. And the Schwann cell, neurolemma, remember, like sarcolemma is the membrane. This is the neural membrane, but just learn Schwann cells. They are on peripheral nerves, and they make the myelin sheath. And really, it's very similar to oligodendrocytes. And depending on who does AP2, they may talk about regeneration. The idea, for instance, the cell bodies of the nerves that are here, let's say in your arm, your forearm, are back here by your spinal cord. If somebody's in a terrible accident, like an industry, and it cuts the arm completely in half, and you have it, if it's handled properly and it's a sharp cut and you can get it immediately, we can reimplant that. The whole idea is you can sew blood vessels together, the bone will heal, but the nerves are critical. The two critical pieces are sewing the blood vessels together. The nerves will knit because peripheral nerves, the cell body, which is kind of the nucleus, though we don't use the term for nerves, is up here. And so the nerve will not deteriorate, it'll grow back together. I had patients, and normally after post-operative, you make a cut, that area will be numb. But because you're going to cut through nerves, you can't help it. Okay, we're operating on bone, you're cutting through nerves and other joints, other tissue. They come to me and they say, everything went great, but I don't like the numbness. And I'm like, we can try this and this. I can't guarantee. I told them beforehand, you're going to have patchy numbness, most likely. And they came back a year later and go, it's a miracle. My sensation, that's because the nerves will go back together. It's not my brilliance, because I was anything but a brilliant surgeon. I was just a pedestrian type of surgeon, average. Peripheral nerves, this is a sensory nerve. You're going to learn that it's called unipolar. The nerve, these are the sensors, like say in your skin, hot, cold. And it's carried along here. Here, look at the Schwann cells. They only are, you have multiple cells, not on multiple nerves, but one at a time. The nerves are larger. The Schwann cells wrap around them. We'll see how that works down the road. And here they have the same gaps. Again, nodes, Ranvier or myelin sheath gaps, doesn't matter. And this is the cell body. That's the nucleus. But we use the term cell body or perikaryon or soma because clusters of these in the brain are called the term nucleus is already used. You learned about parts of the brain. You learned about the basal nuclei. You learned about the red nucleus, nuclei of all the cranial nerves. They are clusters of those cell bodies by definition. If they're outside, clusters of these are called ganglia, the singular being ganglion. The sensory nerves, and we're going to see them when we look at the spinal cord anatomy after this, not today, they're called dorsal root ganglia because they come in the back dorsally. They're all your sensory nerves. If you didn't know it, if you go to microbiology, we talk about herpes viruses. Once you get a herpes virus, you have it forever. It doesn't mean it's never going to bother you again. It just doesn't go away. It becomes latent. This is where it hides in sensory nerve, dorsal root ganglia. So it's anatomically relevant to disease. That to me is very important. And about 1/3 of us, it'll come back after that happens. And it's got a very famous name for the disease when it shows back up. It's not herpes anymore. It's called shingles. Don't have to worry about it. 1/3 of us are going to get it. It's okay. That comes from a different kind of herpes infection. That's really what happens after chickenpox. Okay. And as you'll learn in micro, everybody's been exposed to chickenpox. Because if you had the immunization, you were exposed. There's enough of it going around that even if you didn't get it, you were exposed. Or if you're a kid like me, we all got it. So the nerve cells themselves are the structural unit. They're very specialized. You have them lifelong. They don't replicate. I don't even know of the exceptions. They require continuously amounts of oxygen, continuously a supply of oxygen and glucose. The brain only works with glucose and only works with oxygen. If you take away somebody's oxygen for 3 to 6 minutes, they're going to die. Their brain's going to die. Unless they fell in a frozen pond, then you got a chance. That's well known. Glucose is a big thing. The big disease to know a lot about is diabetes. Diabetes it's typically characterized by irregularities in utilizing sugar, by a high sugar that has to be dealt with in some way, shape, or form. It's really sugar volatility. If you have a diabetic and they have high blood sugar and they either can't correct it or don't try to correct it or modify their diet, blah, 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 whatever it is, they're going to die, albeit slowly. If they have low blood sugar, they will die almost instantaneously if it's not taken care of. Your brain will shut down immediately if it does not have enough glucose, and no other sugar will do. Typically what happens, I've had patients this happen to, they're in a hurry going to work, they're insulin dependent, they're brittle to start with, take their insulin, oh man, I'm late for work, it's only 10 minutes away, I'll eat when I get there. You're supposed to eat right away after taking your insulin to avoid this. They end up with a massive amount of low blood sugar, glycemia. This happened to one of my patients. He goes, I did this, and he said, I remember parking my car at work. It was 10 or 15 minutes away. The next thing I knew, I was waking up in the hospital. They had given him a shot of sugar, which is the way you treat them. Any diabetic, whether it's low or high, because if it's low, you got to correct it right away. If it's high, you can always get it back down with insulin. And it's like, where am I? I'm fine. And you're perfectly fine. It's like, where am I? What happened? It's like you woke up and nothing happened at all. You can move. There's no problems. It's like you got up from a nap. The reality was your brain was starved of glucose, and as soon as you reached a critical level, it shut down. And he was fortunate that he didn't lose consciousness when he was driving, hurt himself, or potentially hurt or kill other people, as well as himself. Because you're lucky if you got to park in the parking lot or pulled over that you felt, oh my God, you got to the side of the road, stop the cars, knocked out. Be very careful. Diabetes is good to know a lot about. The cell body, also known as is really the nucleus. It's really where everything happens. It's where the stuff is made, all the passageways, the neurotransmitters. There's a lot of pigment. You can see the other terms, perikaryon or soma are there. Typically, the neurotransmitters, when they're made, it's just like when you saw the red nucleus or the substantia nigra. It's not unusual to see pigments that are there. The plasma membrane is the big player, receiving the information. That's really where it's when those contact points, they trigger that membrane, and that really sends the signal. It's probably the way they should have said it. Again, clusters of those cell bodies, the nuclei, the individual nerve cells, as long as they're in the central nervous system, they are called nuclei. That's why we can't use the term elsewhere. And ganglia, like I was showing you outside. And this is where we stop. This is where I stop with the other. What I'll be talking about in a recorded version, I'll be identifying these parts. The receivers called dendrites, the transmitters called axons, which we did a little bit more about this stuff. We'll look at different kinds of potentials. That's what a nerve actually looks like, incoming and outgoing. You'll see how long they are. We'll talk about how materials are moved around, and we'll talk about the myelin and how it's rolled around, and the sheath gaps, and all that stuff. And you see, we'll go over a lot of this stuff again, the different kinds of nerves. Okay? So that's what you'll be getting. That'll be my next presentation for Friday, so look for that. Thank you. We are done.